Hi everyone, I am here to talk a little bit about my project today. So I'm doing my PhD with the University of Sydney and with the Marine Invertebrates Department here at the Australian Museum. And recently I was lucky enough to join the scientists on board the RV Investigator, which I thought was interesting to talk about as it gives a bit more context into how especially the marine specimens end up in the museum. So this gives a bit of context to the journey that we took. So we took about 5 weeks to travel from Darwin out to the Cocos Islands and the surrounding seamounts. And we finished up in Perth after about 11,000 kilometers of the voyage. And the main objectives of the voyage was to map the bathymetry of the seamounts. And you can see them dotted through the track there, where it kind of goes green, yellow, red. And also to survey the faunal communities and get an idea of what kind of animals are living on these seamounts, as they're very unique habitats away from the continental margins. And it was important to do this because these two areas, the Christmas Island, which is top right, and Cocos Island on this left side, have recently been designated as marine parks. So we need to know what's there. These are some really cool images developed by the mapping team on board the Investigator, and I thought they really show the kind of diversity of the environment of the different seamounts. So as you can see, some of them are kind of narrow at the top with these deep ravines either side. Then you've got another one which is kind of shallower on top with these interesting cone features, and then some that are really large with these really broad flat tops. And we also were able to gather this data on the density of the seabed, which was really helpful in for the team deciding where we deploy the trawls. So you can see the lighter shade is where the sediment, the density is less, and it's like soft sediment. And on the sides, you can see it's very rocky, harsh environment that you don't really want to put a net down. So that really helped us to work out where to put the nets. And you can see them on these images. The blue bars shows where exactly we deployed a trawl. And in total, we did 47 trawls over 22 days, and we surveyed from 160 meters down to 5,400 meters. Of particular note was this seamount called Murfield, just because of its grand size, but also the fact that it's comes to a really shallow depth at its peak. So it rises from about 4,500 meters to within 17 meters of the surface, and it's about 60 kilometers across. So it's very huge. And we ended up deploying 15 trawls on this seamount from 160 to 4,000 meters. And that's really useful to get a really broad depth range of specimens in one given locality. And just thought I'd give a bit of context to life working on a research vessel. So this is the lab, a little bit different from what we're used to working in, but it was super fun. So the specimens come into this lab from the back deck directly out of the net in big crates of ice. And we spent lots of time here sorting them, labeling them. We had a really good photographing station, so we photographed every specimen as we go, and then they get preserved, ready to be transported back to the museums and go straight into collections. So a very efficient process through this onboard lab. And here you can see a nice array of lots of the frequently sampled groups that we got of invertebrates. So we got lots of hard corals, crustaceans, sea cucumbers, echinoderms, corals, all sorts. And also not forgetting the worms, as this is what I'm studying. So yeah, a nice diversity of worms as well. And particularly this one on the middle left is a scale worm, and that's the genus and the family of worms that I've been studying for my PhD. So I'll just tell you a little bit more about those now. So my research so far has been looking at the diversity of scale worms along a bathymetric gradient. I've been looking at one specific genus, which is called Laetmonice. And I've included a few rather messy specimen images of the ones that I've been working on so far. And you can already see kind of a bit of morphological diversity across these specimens. But my research was interesting because all of the specimens that I've included in my research were preliminarily identified as Laetmonice uramba, which is, I've included a photo of the holotype that we have here at the museum up there. And they're an interesting group study because they're found in abundance from shallow waters to deep. But given the huge depth range of my data sets, I wanted to see if there is evidence for this species having Eurobathic distribution. So it's one species found in both shallow and abyssal depths and all those in between. And it's interesting to note that the type locality of uramba is in Botany Bay here in Sydney in shallow water. And also because I have a data set from around Australia in different depths, the logical question is, are they all really Laetmonice uramba? So this shows the extent of my data set as it stands. I have 80 specimens, all stored either here at the Australian Museum or Museums Victoria. They've lent us those. And they've all been sampled on the RV Investigator between 2015 and 2018. So that was really good for me to have that experience to see kind of where my specimens have come from. And the ones I have so far are all from the south and east coast of Australia, from a huge depth range from about 200 meters down to 4,800 meters. These are the results of the phylogenetic analysis that I've already carried out using CO1 and 16S markers. And it shows kind of what was expected from this group of 80 specimens, that it's not just one species, it's a species complex. And we found about 9 different species from these results. And they're organized into 2 main clades. First, you have this large clade at the top, which I'm keeping named as the Laetmonice CF uramba clade, given that the specimens in this clade bear the closest resemblance to uramba, and also are most closely sampled to the type locality of uramba. And then we have another clade lower down here, which is the Laetmonice producta clade, and all of the Tasmanian specimens are grouped there. And then we have a couple of odd ones out down here, which are a bit of a mystery at the moment. But the results kind of confirmed what we were expecting. So the main finding is that, yes, they are all distinct species, not just one species, and they're all going to be currently undescribed species, but also that they are organized by depth, which is kind of what we were hoping to prove with this awesome data set from such huge depth range. So all of the shallow specimens are all closely related, and similarly down the deepest ones are more closely related. And we also found that the deeper species, so for example, clades 7 and 9, also demonstrate quite a large depth bathymetric range, which is also to be expected of deep water species. So yes, in conclusion, these results have confirmed that all of these specimens around Australia that we found are distinct species with each their own specific depth range, and we did find large bathymetric range in the deepest species, and also the greatest species richness around 1,000 meters. So 5 of the 9 species in my tree were found at around 1,000 meters, which is to be expected as there's greatest kind of environmental heterogeneity around here, around that depth. And so the next steps of my research will be conducting a thorough morphology study to see if there's any morphological differences in the specimens that align with these species boundaries. And I'll be adding some more samples from other localities and depths. So for example, the voyage that I just went on from the Indian Ocean, and they've also just concluding a voyage along the west coast of Australia. So I need to check in with them to see if they've got any scale worms that I can add to my data set. Thank you.